Welcome aboard the System76 transmission log. Our broadcast is about to begin. This is the latest on System76 computers, manufacturing, and Pop OS. Now for your in-orbit crew.
Hello, everyone. Welcome back to the System76 transmission log. I'm Alex, our resident normie.
And I'm Emma, wondering why we're doing the intro when it was supposed to be the banter section, but whatever.
I mean, intro always comes first, right?
No, I feel like we always do banter first.
Well, in the end, the intro comes first.
All right.
Just as a movie magic secret, we're actually doing this last. We're not. We're doing this first.
Oh my God. Just like trying to confuse me.
So I did want to point out I'm wearing pink today.
Oh.
I came prepared for our hosting here.
What's the rest of that shirt?
Oh, the rest of the shirt is it's Slither's Snakeman. It's a snake disguised as a human.
Is that a cartoon?
No, it's just a shirt I found on the internet from a cartoonist.
Mm-hmm.
But I was curious about if we could get into some Emma lore here. When was like the first moment you were like, oh, pink, that's my jam now?
Well, when I really embraced it, I was probably like 15 or 16. I mean, I loved it when I was a kid, of course. I was really into Barbie, and I just think pink was the best world to live in. But yeah, when I was 18 with my first apartment, everything was pink. There was a pink pillow floor area. There was a bright red couch and a pink table, and everything on the walls was pink. The dishes were pink. And ever since then, I was just like collecting pink stuff. And slowly, pink clothes have been making a comeback. It was really hard to find a pink shirt or a pink dress for a really long time. But now I can pretty much find that stuff anywhere.
Do you have a favorite shade of pink?
I like a berry type of pink. It's not quite hot pink. It's not dark. It's like in the middle.
It's like the inside of a strawberry.
No. That's pretty light pink, I think. The color of my shoes is a very good pink.
Okay, that's like a hot pink, I would say.
Well, I like it a little more subtle, but can't choose them all.
True.
All right, I have another topic to discuss really quick.
What's that?
The balloon game.
Oh, yeah. I wasn't there for it, but I hear it's pretty poppin'.
I just realized that you weren't there for it, so how are we going to talk about it?
Well, I mean, I know. First of all, what is the balloon game?
Oh, I'm glad you asked. So everyone at the company in the factory at the beginning of the day gets a balloon with helium. And at the end of the day, whoever is left with a balloon gets a prize. But throughout the day, people can hide their balloon, pop people's balloons, or steal people's balloons. So we had some really competitive people, had some tricky people hiding them. I thought Young Chin did a great job hiding his behind the curtains in the window because you could totally see it outside, but you couldn't see it on the inside and nobody found it. So he was one of the people left standing. And then Josh, I went and had a conversation with him while he was in the shipping container, asking him if he had seen any stray balloons. Turns out his balloon was in the top corner of the shipping container while I was talking to him. And I ended the contest and I was like, congratulations, everybody. And then he comes in at the last second and is like, I still have mine. And then I asked him to expose his spot and it was right there. And I couldn't believe it. So I am not very good at the game, but I thought those two spots were very clever. And then we had Grace, who got all the balloons. She was taking people's balloons and popping them left and right. And she came in my office at the end of the day and pulled out her balloon from under her sleeve. And She was very proud. So she got the most. Young Chin and Josh were probably the most clever about it and sneaky. But all around, it was very fun.
How did she fit an unpopped balloon up her sleeve?
Very carefully. She wears pretty baggy clothes, but her balloon had been losing air throughout the day too, so it was a little smaller than the other balloons.
Mm-hmm.
But yeah, I think she called it George or something. She named it.
Well, we're glad George survived. What would you say was the best technique that was used for popping these balloons?
I didn't witness any popping. I only heard it. But every time you heard a pop, you heard a giggle too, which was really funny. No matter
they-
where you were in the factory, you could hear somebody laughing about it.
So the balloons weren't just filled with helium, they were filled with laughing gas.
They were filled with laughing gas, yes.
Or possessed by goats.
Mhm.
Ghosts, not goats.
I'm pretty sure
Possessed by goats.
Mm.
I said it again.
Well, what?
We're leaving that in.
Yeah, that was really fun. I think we got to do it again, though. But I have some different ideas. Like the person with the most at the end should probably win an extra prize.
They have to throw a dart at a wall of balloons that each contain a prize in them.
I like that.
Yes.
I like that a lot. Okay, well, now we know which games to implement in the future, but maybe we should get into the news.
Yeah, let's get into the System76 lore from the past month.
Okay. Well, it's kind of the opposite of last month because we had a ton of news.
Went from lots of releases to gearing up for, some releases down the line.
Yeah. So this month we have the tiniest bit of news, and it's about the smallest bit of hardware, the little Meerkat.
He's the little baby with big biceps.
It's our compact mini little desktop in the Nook form factor. Fits right on your desk or mounted on the back of your monitor. And it is rocking some Intel Core Ultra CPUs with up to 4.8 gigahertz processors and a whopping up to 96 gigabytes of RAM and up to 16 terabytes storage. So much in such a little package, still rocking Intel. But also we added...
I can't read your lips there.
I thought you would just know. I thought you were going to be like, open firmware.
Oh, yeah. Look, I'm one of the first stages in the product release. Like, once the product is ready to go, I absorb all the information about it. I output it into something for the website. And then, my mind is cleared of it because I'm on to the next thing.
Oh.
So
what you're saying is you forgot that Meerkat has open firmware.
It's not that I forgot so much as I've stopped thinking about the Meerkat for like the past 3 weeks.
Okay.
So it's been a while for being able to spout off the highlights, which is what we're here for on this podcast. So yes, Meerkat now has open firmware. It's our core boot based System76 open firmware. Part of it is speeding up boot times, minimizes the firmware so it's more secure, less vulnerability, all that good stuff.
Yep.
Do you think if you tied a balloon around a Meerkat, it would float?
That's a great question.
We should try that.
Well, I'm pretty sure one balloon won't do the trick, but we should see how many balloons it will take to float a Meerkat.
My guess is like 3. Helium's pretty strong in a balloon.
Okay.
They floated a house with it, so.
Yeah, but he used like 1,000 balloons.
He had a lot of balloons.
Yeah.
It was a big house, though. You do like a townhouse, that's half the balloons.
Yeah. Meerkat, since it's just a little Meerkat, probably 3 or 4, yeah.
With no mirror potential.
Well, I'm going to test it and we'll come back to you next month on the podcast and tell you how it went.
Okay. Yeah, we got to test it for science.
Yep, I need to know. All right, well, that's our hardware news.
We have some community news, right?
We do. We are going to be at a couple conferences in March and also April and May. So to start off in March, we'll be at Scale in Pasadena, California, which is March 6th through 10th. And we'll be hanging out at our booth along with our friends at Ampere, who will be next to us. And we'll have 2 Thelio Astra demos at the Ampere booth showing us all the cores, all the power, all the Blender renders you can think of.
And a driving simulator as well, I think.
Yeah, Tier 4's driving simulation that they had at CES will be displayed. We'll be doing the same simulation at our booth at Scale at the Ampere booth. And then also on the other side of the booth, we'll have a cosmic section where you can design your own universe and then see your cosmic theme in action. And then also we'll be giving away 2 launch keyboards and a lot of swag. And you can get your hands on some hardware and talk to some of your fellow nerds.
Yeah, Garrett, who's on our QA team, he worked on a really cool universe designer using the cosmic theming system. So we're really excited to show that off.
Mm-hmm. And then also in March, we're going to be at the PNY booth at GTC in San Jose, March 17th through the 21st. PNY will be showcasing the Thelio Mega and just kind of showing off what a high-end workstation optimized for multi-GPU compute and heavy graphics and peripherals would be performing. So
yeah, subject to change potentially, but what we've been told is there will be a demo showing off some new stuff, and it'll be using a production workflow, a combination of Blender, Unreal Engine, and OBS. So all tools available on Linux, but just showing off the power of our hardwares combined.
Cool. That one's going to be a cool demo.
Yeah.
And then let's see, April, we have Linux Fest Northwest, which is my favorite one of the year. That one's in Bellingham, Washington, the end of April. And we'll be doing a talk on how far Cosmic has come and also have a booth. And then we'll be hosting the Saturday barbecue.
I could use some barbecue right about now.
It's actually not really barbecue. It's just like whatever food is a good idea that day. But maybe just hot dogs like last year.
If you bring some barbecue sauce, it's barbecue.
Okay, that's fair.
Join us with your barbecue sauce, BYOBS.
Bring your own BS.
To the Linux Fest Northwest barbecue.
Hope to see you guys there. All right. What's happening in the cosmic universe?
Cosmic universe. Let's see. There is potential for delaying a week to get some more features into the Alpha 6 release. So I could say some stuff that's potentially wrong. An easier way to go about this is just head to the blog for actual up-to-date news, blog.system76.com. But what I know and I've heard inklings of is we now have desktop icons. So you right-click the desktop, you go to desktop view settings, and that'll bring up some toggles for showing desktop icons, showing the trash icons, adjusting the size and spacing, things of that nature. There's also some more polish updates to the way workspaces functions. Multi-monitor setups, you can now drag and drop windows into the other workspace overview on another display. You can also drag and drop minimized windows. When you drag a window out of a stack to another workspace, it'll now only move that window like you'd expect. I think previously it would move the whole stack. So, there's more updates along those lines just to make workspaces feel polished and intuitive. There's file menu features built out for the Cosmic Media Player and potential for the initial setup applet, OS update settings, and a desktop zoom feature for accessibility. Those things may or may not have been released. Check the blog for accurate information, but if not released in Alpha 6, probably by Alpha 7. So, all that said, exciting things for Cosmic. I very much enjoy using it. I think I'm still on 2204 LTS, I've learned. But even being there, it's been working great for my workflow. I don't really get into the nitty-gritty. I'm more surface level, but it's been going well. I have seen that the compositor on 2204 LTS will no longer be getting updates, so make sure you're upgraded to 2404. I think the ISOs are 2404, so you should be all right.
Thank you for that piece of advice.
Yeah.
All right. Well, we have a very interesting interview today.
Yeah, we have RJ Nowling here to talk about his work in genomic research.
And it's been really bugging me all week to get this interview out there. So here we go.
My name is RJ Nowling. I'm an associate professor of computer science at the Milwaukee School of Engineering. I'm also the program director for our master's in machine learning. My research is focused on what I call genomic data science. I work with biologists to process their raw sequencing data into a usable form and then analyze it using data science techniques. So in many cases, the data we're collecting during an experiment, there's a particular research question in mind. And often I'm helping them determine what evidence the data might provide to help answer that question. Earlier, you mentioned you looked at my website and you saw insects. Yeah, I primarily work with insect genomes. A lot of my collaborators work on mosquitoes such as Anopheles gambiae that vector malaria, as well as Aedes aegypti, which is a mosquito that vectors dengue fever. So these are like medically important organisms to public health.
So it's mostly for public health that you're doing all of this, or are there other reasons?
So I'm doing it mostly from a pure biology aspect. I want to better understand the biological systems. And we hope that by studying these insects, we'll also learn things that are transferable to other organisms. But it so happens that the biologist I work with may have more specific questions. Like, for example, one of my collaborators is trying to better understand why some of the Anopheles mosquitoes, their immune system can fight off the malaria parasite and some can't. Like, even if they're from the same family, literally like the same mom and dad, there's differences. And she's trying to understand the genetic basis of that. On my end, most of the work I'm doing isn't necessarily specific to the mosquito. The data I have is specific to the mosquito, but the techniques I'm using are general.
So in the case of malaria, that could lead to breeding mosquitoes to eventually,
Yeah.
if there's a gene associated to eliminate the vulnerability to malaria.
Yeah, you're absolutely right. So there's been some experiments, what they call gene drive, to try to basically produce sterile males and such. Other groups have been developing techniques to like, okay, can we release some genetically modified mosquitoes into a population and hopefully they will basically take over that population and now most of the population will no longer do a certain thing or something.
Mm-hmm.
So in this particular case, yeah, if we can find what makes it so that some of the mosquitoes, their immune system can't destroy the malaria parasite, we can then use gene drive techniques to basically ensure that the mosquitoes can fight off the malaria parasite so that they don't transmit it to humans.
That's very interesting.
Yeah.
How did you get into this?
Actually, I was exposed to bioinformatics through a summer internship arranged by a family member. So when I was in high school, I interned at the University of Connecticut Health Center, making a simple website with some data from a paper. And I really fell in love with it. I loved that I could apply my computing skills to understanding something in the real world. And I liked the nature of the work itself, like data processing is interesting to me. So I made it my focus in college, and then I went and got a PhD. And so during college, while many of my colleagues were doing internships with companies, I was doing research with professors. I did spend a couple of years in industry after my PhD. So I was at Red Hat for 2 years, and then an online advertising company called Adderall for 2 years. I started my faculty position at the Milwaukee School of Engineering in fall 2018.
And when along that journey did you discover Linux? Was it at Red Hat or was it before then?
It involved Red Hat, but it was not while I was at Red Hat. So it's actually in middle school. Someone gave me a box copy of Red Hat Linux 6.2. And I want to clarify, I did not omit the word enterprise. This was before it became Red Hat Enterprise Linux. This was old Red Hat. And so if I remember correctly, like the version I had ran GNOME desktop version 1.
Oh, wow.
And so then I started using Slackware some in high school, and I later switched to Debian. And so now all my research machines, I use Debian for all my workstations and servers, basically.
Mm-hmm.
And then my laptop and desktop, I use Apple products.
With regards to your workflow, what software tools are you using mainly?
So when we get the raw data, we clean it with something like TrimSeq, and then we align it to genomes using something like BWA. We'll use a software package called GATK to try to identify where individual samples differ from a genome. We might also use, depending on if it's a different workflow, a tool called MAX for what's called peak calling.
Mm-hmm.
So there's certain experiments that basically highlight certain parts of the genome as like this part of the genome has activity of interest. And then once we get have produced that sort of output, then we can switch to more traditional data science tools like using pandas and Jupyter notebooks and, scikit-learn and things like that.
I see.
For your desktop, I know you have a Thelio Astra. How has working with Thelio Astra affected your workflow and your project?
It's massively increased my throughput. So the data sets I work with have a lot of samples, right? So the biologist will sequence like the DNA from 1,000 mosquitoes. Well, it's hard to get much parallelism for an individual sample, but the samples can be processed independently. So the ability to have 120 cores instead of 24 cores means I can process a lot more samples at once. It also tends to be very memory intensive. So the fact that I can have 512 gig of ECC RAM is also an important component of scaling up that pipeline.
Mm-hmm.
And these pipelines often take weeks to run. So it can cut it down from a month to a week or a few days.
Wow, it's a big improvement.
Yeah.
What led to your decision to go with System76 and the Thelio Astra?
Honestly, it was price. So I've been wanting an Ampere Ultra system for a while because like I had a workstation with an AMD Threadripper processor in it, 24 cores and 120 gigs of RAM that I got 5 or 6 years ago. And that's been my main research machine. It was time for an upgrade. But whenever I would look at other systems with AMD Threadripper processors, I just couldn't get more cores or much more memory within my budget. The Ampere Ultra platform offers a really nice price point for getting a lot of cores and a lot of RAM at the same time. And I need both to do my work. And System76 was the only company I could find that really was supporting a workstation option for Ampere. And, I can say also that I'm glad I went with System76 because support has been super helpful. I haven't run into any major problems. It's just my ignorance. So I didn't realize that because it's running a server motherboard, that it takes a really long time to post before the boot screen shows.
Mm-hmm.
So I thought something was wrong with it when I got it, when it's like, why isn't this showing anything after like 5 minutes? And it turns out I just needed to wait longer.
Oh,
But, the support was super helpful and I really appreciate my interactions with the company. I think that has potentially turned me into a repeat buyer in the future.
that's awesome.
Yeah, support's been a long time pride point for us, for sure.
Do you have advice for people who want to get into genomic data science?
Yes. So unfortunately, most of the people I know doing this are at universities. There are some companies in industry like Illumina, which provides some of the sequencing technology, also hires a number of bioinformaticians and data scientists and software engineers, but still primarily most of the people in this field are in universities. So you pretty much need a PhD. That said, I have worked with people in PhDs in a wide range of fields from computer science to biology to statistics, physics, chemistry, applied math, whatever, that we all work on these projects together. And so I think one thing I had to figure out is that you have to pick a home field. So in my particular case, I'm very comfortable teaching computer science and data science courses. So getting a PhD in computer science was the right fit for me, even if my research itself is very interdisciplinary. And so the trick is to find a faculty member who's working in like bioinformatics who can advise students in a PhD program that you would be successful in, basically.
Mm-hmm.
And for college students, you need to start talking to your professors and seeing if you can get opportunities to do research with them as an undergrad. That'll tell you if you like the work, and that's how you build up your resume to increase your chances of getting accepted into a PhD program.
That's awesome advice. Thank you.
Yeah.
Looking forward to the future with machine learning and genomic data science, how can you combine those in the future to make innovations?
Yeah, some of the work that I find most interesting recently. One thing to know is our genome doesn't just have genes, it has other non-coding elements that play an important role. So one example of these are called enhancers. Enhancers help determine how many copies of a gene are produced. And we're actually just learning that a lot of diseases are caused not by changes in the gene themselves, but in the enhancers. So your body just may not produce as many copies of a gene. So one thing, though, is that the enhancers are hard to find computationally. Generally, people use experimental techniques, but there's been some really interesting work lately using deep learning to identify enhancers in an accurate mannerism. So because it's finally pretty accurate, we could actually go through all the publicly available genomes of the NCBI database provided by the NIH and find the enhancers for all of them. And then that would allow us to do cross-species comparisons. Another thing is that enhancers are generally not located near the genes they interact with. They're pretty far apart. So there's been some work to try to computationally determine which enhancers act on which genes which will help us better understand what genes they impact and thus what potential diseases. The other thing is trying to understand how genetic variation within the enhancers has an impact. Sometimes the DNA changes and it does nothing. Sometimes it increases the number of copies of a gene. Sometimes it decreases it, but it's hard to predict. So all of these things, if we can build accurate machine learning models to do this, we can vastly speed up research and reduce costs and study organisms that we can't study right now because it may be infeasible to do the actual experimental lab work for whatever reason.
Mm-hmm. Is that what you're most excited about for the future of your field?
I get really excited when I can understand the mechanisms of the biology. So in some ways, I'm a biologist who uses a computer as my instrument. So I'm excited for advancements that will allow us to better understand how the biology works. That's my main motivation.
And how can people get in touch with you if they want to learn more or connect with you?
They can check out my website, rnowling.github.io, or they can send me an email at nowling@msoe.edu.
Awesome. Is there anything else you'd like to add or share with the audience?
Just that I've really enjoyed my experience with the Thelio Astra so far. It's been a great machine and worth every dollar. And it really does offer a lot for the price. Highly recommend it.
We're so happy to hear that. Thank you so much.
Yeah.
We really appreciate you coming on to share your experience and your story and your work.
Well, I'm happy to be here.
All right. Well, that's our, well, no, it's not our show because we have a game.
Ooh, we have a game.
Yes.
What's our game?
Okay. In the spirit of the balloon game, I got here first. As you know, I always get here first. I hid a balloon in this studio and you have to find it. But you only have 30 seconds.
30 seconds? Am I able to see it from where I'm sitting, or do I have to get
there
up?
There are no hints. I don't know why you're asking questions about hints.
Well, I'm asking because I have headphones on. Do I have to take them off and get up and walk around?
Yes.
Okay, it's going to take me 10 seconds to get out of this chair.
Hold on, I got to start the timer.
Can you start it once I'm out of the chair?
Yes. Okay, ready? Stop looking. Okay, go.
Not under the piano.
Looking under the piano, not there.
Not behind here. Is it in your jacket?
No. 5, 4, 3, 2,
I found
1.
it.
No, you didn't.
No, I didn't.
All right. Finally, I stumped you on a game. I feel like every single podcast you win. So I really needed a win here.
What's your prize?
Pride.
Oh.
I'll show you where it's at. I have to get out of the chair.
You only have 20 seconds to find it. I even looked there.
I know. That's why it was so funny. All right.
It was between two large foam boards. I don't
like
It's
sound.
like sound suppressant boards,
Sound blockers.
but table sized.
All right. Well,
I'm not saying that.
I'd say good job, but you didn't do good that time.
No, but I won a balloon.
All right. Well, thanks for joining us today, guys. We'll see you next month.
On the System76 transmission log.
Over and out.
3, 2, 1, 0.
This has been the System76 transmission log. For more inspiration, check out the website and follow us on social media. On your descent back to Earth, please keep your hands and feet inside the transport beam at all times. Captain Sign-Off, in transmission.